Michael Lee, and this video is about genetic engineering and genetic editing. To start off, let's learn some basic genetics, because if you don't understand the science of genetics, then you can't edit DNA. Deoxyribonucleic acid, or DNA, is a molecule that consists of two strands, the template and complementary strand, and is thus a double helix. It is located within the nucleus of every cell except for red blood cells and mature hair and nail cells. It contains the genetic information needed to produce proteins for the cell. The DNA strand consists of a backbone and one of four bases. The building blocks of DNA, which consist of a part of the backbone and a base, are called nucleotides. The backbone of DNA consists of phosphate and deoxyribose sugar. The four bases are adenine, thymine, guanine, and cytosine, which are represented by the letters A, T, G, and C, respectively. There are also rules that dictate what pairs of bases can occur, called base pair rules. Adenine always pairs with thymine via a double bond and is larger or more complex than thymine, while guanine always pairs with cytosine via a triple bond and is larger or more complex than cytosine. This is important to know for genetic editing, because if you don't understand the structure of DNA, then you can't understand how genetic editing works nor use genetic editing. For example, if you don't understand that G and C are paired, then if you see G and T paired, then you might not edit the DNA to remove T from it and replace it with C. If you don't know what the base pairs are in the DNA and what they are, that they are part of DNA, then you are editing blind, as you literally don't have the information necessary to fix mistakes in the DNA. Central dogma, also known as protein synthesis, is the process of turning DNA to RNA to protein. The first part is transcription. Transcription is the process of creating RNA from a section of DNA. The enzyme RNA polymerase first cuts or unwinds the section of DNA that needs to be transcribed. Then it puts nucleotides with a backbone consisting of ribose sugar and phosphate that have a base matching the bases of the template strand in the nucleus to create a strand of RNA. The RNA leaves the nucleus and travels to the ribosome in the cytoplasm of the cell. When the RNA reaches the ribosome, translation takes place. Translation creates proteins from the instructions on the RNA. On mRNA, a group of 3 nucleotides is called a codon, and translation starts once the start codon AUG is found and stops at the first stop codon. Stop codons are codons that tell the ribosome to stop translation. Transfer RNA or tRNA transports amino acids, the building blocks of proteins, to the ribosome. Each tRNA has a set of 3 nucleotides called anticodons, and these anticodons match the codons of the RNA. So the amino acids are brought in order of the codons on the mRNA. What amino acids match which codons are already mapped in a codon chart. At the end of translation, the amino acid chain separates from the ribosome and folds into a protein, with the specific protein being determined by the length and order of the chain. If you understand this process, you may not understand why and how a mistake in DNA would result in a change in the eventual protein. For example, if you have a codon chart in a DNA section, is supposed to make the codon that makes the amino acid pair in the RNA, but the DNA section in the DNA you're editing makes a codon that makes the amino acid sir, then the protein is entirely changed. However, if you don't understand central dogma, then you wouldn't know there is an error, because the codon chart has codons for mRNA, not DNA. Mutations are errors or mistakes in the DNA that occur randomly. There is always a natural chance that they will occur, but certain chemical and other environmental exposures, such as radiation, can increase the chance of mutations. There are 4 main types of mutation: substitution, frameshift, nonsense, and silence. In the substitution mutation, one base in the nucleotide is swapped for another, such as A to G. Substitution mutations only change one amino acid in the chain. In a frameshift mutation, the DNA sequence is either moved to the left or the right, which completely changes the codon of the produced mRNA, causing the protein to change entirely. Frameshift can be broken down into two categories: deletion mutations remove a nucleotide or a paired nucleotide, causing the sequence to move to the left, while destined mutations add a nucleotide or a paired nucleotide, causing the sequence to move to the right. One example is a deletion mutation in the CFTR, or cystic fibrosis transmembrane conductor gene, that causes cystic fibrosis. Another example is an addition mutation in the hexa gene that results in Tay-Sachs disorder. Nonsense mutations add a stop codon that makes translation stop prematurely, which makes the protein shorter. Silent mutations are substitution mutations that do not change the resulting protein. This is important to know because if you don't understand the mutations, then you may edit the DNA incorrectly. For example, if there's a frameshift mutation, more specifically a deletion mutation, and you don't know about frameshift mutations, you may not know how to edit the DNA when there is one. Clustered regularly interspaced short palindromic repeats, or CRISPR, consists of two parts, Cas9 and guide RNA. Cas9 is the protein that cuts the section of the DNA, while the guide RNA tells the Cas9 which section of the DNA to cut by finding the section that matches the guide RNA. In order to conduct genetic engineering, a guide RNA must be made from the mutated DNA and then introduced into the body of the person you want to edit. Then the cutout section of DNA must be edited to what you want it to be and put back into the DNA. Genetic engineering can be used to treat genetic disorders such as Tay-Sachs disorder and cystic fibrosis. It can also be used to cure heritable, non-lethal conditions that often impact people's lives, such as color blindness, myopia, and deafness. Genetic engineering may even be used to help prevent infectious disease by making the body produce immune cells from a young age that fight off HIV and malaria. However, genetic engineering can also be used to make bioweapons, pathogens such as bacteria and viruses that already cause humans to be sick, edited to be deadlier and more infectious than the disease they usually are. They may be used to engineering kids to be unhealthy, such as giving them deafness or intentionally giving them genetic diseases. Some people have Munchausen disorder by proxy, a form of Munchausen disorder, a disorder that usually causes them to pretend to be sick for care and attention, that causes them to make other people, usually ones that they care for, sick, or pretend that such people are sick in order to get care and attention, and may use genetic engineering in order to make their kids have chronic genetic diseases that will make them sick. It may also be used to engineer super soldiers for bad people. For example, the supreme leader of North Korea, Kim Jong-un, may decide to use genetic engineering to make physically stronger soldiers in order to defeat South Korea and thus unite the Korean Peninsula under an autocratic regime that he would lead. Yeah, Kim Jong-un runs death camps pretty much. If he takes over the entire Korean Peninsula, you're gonna have a bunch of executed South Koreans. Not exactly a good idea. Anyways, these sorts of possible applications have created controversy over genetic engineering. Some argue that genetic engineering is good because it gives parents more options, reduces death by eliminating genetic diseases, and will improve society as humans are edited to be better. However, some argue against genetic engineering because designer babies engineered and selected for by their traits while in vitro may result in genoism, or discrimination towards people that are imperfect based on genetics. Genetic engineering may also be used improperly as a way for autocrats to take over other countries by enhancing soldiers, and may enable the creation of super infectious diseases that would devastate humanity. Personally, I think that people should be able to edit their DNA as long as they understand the consequences of their actions. As if people understand what their genetic engineering and have the means will do to their body, they should be able to edit it. Okay? I believe the criterion for genetic engineering should be informed consent for the participants. Informed consent is the idea that anyone is mentally capable can agree or refuse a medical procedure as long as they understand the consequences of the action. It is one way that bodily autonomy, a keystone of medicine, is expressed. The patient, or if not available, their proxy, such as the parent, should decide whether or not to have genetic engineering done on them as long as they are under medical advice.